Hello, good morning to everyone. Here we are to forget during some minutes COVID and talk about science in our Ciência Falada. Today, our speaker is Cláudia Miranda. Cláudia is graduated in biology and has a master in health sciences by University of Minho. She is currently PhD student in population health science, health research domain of ICBS. And Claudia, if you are ready, you can share the presentation now and start.
You see my screen?
Yes.
Just let me switch this off. Hello to everyone. It's my great pleasure to present here my PhD work, where I explore the crosstalk between immune mediators and cognition in an aging context under the supervision of Joana Palha and Margarida Correia Neves. And due to the increase in healthcare service and in increasing quality of life, people are living longer. For instance, the life expectancy is nowadays approximately 72 years. And this number is expected to increase in the years to come. And as you can see by, let me use the laser. And as you can see by those graphs, the age pyramid structure is changing. The individuals with more than 65 years old represent nowadays approximately 9% of the world population and 20% of the Portuguese population. And it's forecasted to increase in the next 30, 60 years to 17 and to 37 respectively. And this global aging phenomenon is so alarming because not all of us will age in good shape. Some will face age-associated comorbidities and decline in some functions that will affect our daily life. As such, the scientific community has been challenging to join efforts to try to discover which factors are contributing to an healthy aging. And during aging, the human body undertakes several molecular and cellular alterations that affect virtually all the systems in our body, which includes the immune system in a process that known also as immune senescence that affects both the adaptive and innate branches of the immune system. And in the case of the aging of the adaptive immune system, it has been described a reduced repertoire diversity, both in T and B cells. It has been also reported a decrease in naive T cell compartments and a subsequent increase in the memory and effector compartments, and also decrease in the absolute and relative numbers of both B and T cells. On the other hand, the reports showing alterations in terms of number and percentage of cells in the innate immune system present sometimes inconsistent findings. However, it has been consistently reported that their function is altered. For instance, I will give you two examples. In the innate immune system, in neutrophils, it has been reported that the chemotactic and phagocytic capacity decreases during aging, and that, for instance, in NK cells, their cytotoxic function is also impaired. Another hallmark is the low-grade inflammation that is usually observed in the elderly, in a process known also as inflammaging, being IL-6, TNF, and C-reactive protein, the most reported inflammatory mediators shown to be increased in blood of older individuals. Another function that is affected during the aging process is our cognitive function. In humans, it has been described that the speed processing, the working and long-term memory decreases naturally during aging, while the knowledge acquired throughout life maintains stable or even increase. And this impact of aging on cognitive function is not exclusive of humans. It also affects other animals. And older mice present impairments in different memory dimensions, from spatial recognition memory, where aged animals do not discriminate as well the newly displaced object as young mice do, to object recognition and also spatial reference memory, where their performance gets worse during aging, and also to contextual and associative memory. And this aging process is not a homogeneous process. There are several inter-individual differences that can be explained or influenced by several factors. In humans, it has been described that factors such as education, cardiovascular risk, mood, lifestyle indicators, among others, have been associated to this heterogeneous cognitive aging. And interestingly, the low-grade inflammation and also the immune senescence have been described to contribute to this heterogeneity. And similar to humans, in rodents, it has been also described that social interaction, environmental enrichment, diet, physical exercise, these factors contribute to or influence their cognitive behavior. However, the association between immune senescence and low-grade inflammation on their cognitive function are still poorly studied. Even though the scientific community has been accepting that throughout life, the brain, more than being an immune privileged organ, enjoys the privilege of an immune-dependent maintenance. And in fact, several researchers have been reporting that the immune system has the ability to modulate cognitive function. For instance, mice that lack both B and T cells present cognitive deficits, and those deficits can be reverted by the replenishment of the T cell pool in the periphery. After a performance of a cognitive task, for instance, a Morris Watermaze task, it has been reported an increased recruitment of activated CD4 positive T cells to the meninges. And others have also shown that blood-borne factors and plasma from young animals are able to reverse the cognitive impairments and synaptic plasticity observed in older animals. But those evidence associating the immune system with cognitive function are not exclusive from mouse models. In humans, it has been also reported that the low-grade inflammation that is usually observed in the elderly has been associated with their cognitive decline, being, for instance, IL-6 and C-reactive protein, the most reported immune mediators in such association. And we have also reported a few years ago in a healthy aging cohort with 190 participants with distinct cognitive performances that individuals with poor cognitive performance in red present higher numbers of effector memory CD4 positive T cells. And this association is sustained even when we control for factors that we know that can impact this association, for instance, age, gender, education, and mood. And knowing all of these, we hypothesize that in physiological aging, alterations in the blood immune mediators, either cells or molecules, are associated with age-related cognitive heterogeneity. And to test this hypothesis, we defined three main aims that were explored in three independent tasks in this project. And in the first one, we dissected the cellular composition of peripheral blood in male and female mice during aging. In the second part, we determined how the inter-individual differences in cognitive function at old age correlate with the age-associated alterations of the immune system using a longitudinal study design in mice. And in the last part of the work, we determined how the plasma immune molecules correlate with age-related cognitive heterogeneity in healthy senior individuals. In the first task, we perform a longitudinal characterization of the blood immune cell composition using BALB/C mice, both male and female mice. And for that, we collected blood from the tail from 3 to 18 months old, approximately every 3 months. And by flow cytometry, we characterize the main innate and adaptive immune cell populations. In this part of the project, we performed the experiments in 3 independent tasks, sets, and it was comprised in a total of 218 mice in the first time point. And this characterization is extremely important because despite the widespread use of mouse models to study immune system aging and its interplay with other systems, information regarding the blood immune cell composition during aging was scarce. And the longitudinal nature of this work allowed us also to explore intra and inter-individual differences, in contrast to what normally is done in cross-sectional studies, where it's only compared young versus old animals. And to explore the trajectories of the proportion of immune cells during aging, and also to dissect the impact of sex on such association, we perform linear mixed models. And in these graphs, we have represented males in mint green and females in orange. And as you can observe, the overall percentages of the main innate immune cells increase during aging, while the percentages of the main adaptive immune populations decrease during aging, which goes along with some of the findings already reported in humans. For instance, it was already described in humans a decrease in numbers and percentages of both B and T lymphocytes. But despite these overall alterations in the percentage of cells during aging, we also demonstrate a sexual dimorphism. For instance, here in neutrophils, we can see that males have higher percentages than females and also present a faster increase during aging. Another example is the NK cells locking the expression of Li6C, where we observed that females have higher percentages than males and also an almost exclusive increase. And this example is also good to note to notice that the variability in some populations also increase with aging, and that some of the sex-related differences appear or are more evident later in life. In the case of the adaptive immune system, for instance, I will use T cells as an example. We observe that females have higher percentages than males and also present a faster decrease with aging, which was already described in humans, but not in mouse models. And for the sake of time, I will not show CD4, CD8 positive T cells and their corresponding compartments, but on those cells, we also observe age-related alterations in the proportion of cells and also a sexual dimorphism. In the second task of this project, considering the poorly studied association between immune system aging and cognitive function, we decided to explore how inter-individual differences in cognitive function at old age correlate with age-associated alterations of the immune system. And for that, we used the same cohort of mice used in task 1. And besides the longitudinal characterization of the immune system between 17 and 18 months old, we perform an extensive behavioral characterization covering different behavioral domains. To evaluate anxious-like behavior, we use the elevated plus maze test. And in this test, we use an elevated plus shape arena, where we have 2 closed and 2 open arms. And we know that a mouse that is more anxious spends more time in the closed arm than in the rather than in the open ones. For their locomotive activity characterization, we use the open field test. It's an open and empty arena where we let the mice to freely explore. For the depressive-like behavior, we use the tail suspension test. In this test, we suspended mice by their tail and we measure their immobility time. And we know that higher immobility is also associated with a more depressive-like behavior, let's say. And for the cognitive characterization, the main focus of this project, we perform different memory tasks covering different domains. We use the novel object recognition test, the NOR test. And in this test, we habituate mice to an arena, and then we expose them for 10 minutes to 2 equal objects. And 1 hour later, we replace one of the objects, and we then assess their interaction time. And we know, as mice are naturally explorers, that a mouse with an intact memory tend to spend more time exploring the new object rather than the old one. We perform also the Y-maze test. In this test, we use a Y-shaped arena, and we use a 2-trial protocol. In the first trial, we close one of the arms and we let the mice explore the 2 arms. And on each arm, we have spatial cues. And 1 hour later, we open the called new arm and we let the mouse to freely explore the maze. And again, a mouse with an intact memory tend to spend more time exploring the new arm rather than the other 2. We also performed the contextual fear conditioning test, the CFC. And in this test, we measure the conditioned response, the freezing behavior, to a mild food shock in a specific context and also associated with a cue. In this case, we use a white noise cue. And considering that our main focus was the cognitive function and also to reduce the number of variables and to gather all that information, we performed principal component analysis. And as expected, we observed that the percentage of entries in the novel arm obtained in the Y-maze test, as well as the novelty preference, they were grouped in a single dimension that we call spatial memory. The percentage of interaction with the novel object in the NOR test was a single contributor for what we call a recognition memory. And the context and cue discrimination index from the CFC were grouped also in a single dimension that we call associative memory. With the scores obtained on those domains, we perform a key means cluster analysis, and we were able to classify mice as good and bad performers. In these graphs, we have represented good performers in black and bad performers in red. And as expected, mice that have a worse performance in red present always lower scores than the good performance in the different memory dimensions measured. We also checked if the other behavioral dimensions, how good and bad performance were performing in these different behavioral dimensions. And we observed that they have similar behaviors in terms of anxious, depressive like behavior, and also in terms of their locomotive activity. And this is good and also indicative that it seems that those behavioral domains are not affecting the segregation of mice as good and bad performers. And to explore if the immune system profile at 80 months old, last time point, is associated with cognitive performance, we perform different regression analysis. And we include on each model, each of the populations independently controlling for sex. And we observe that the proportion of effector memory, the proportion of CD4 positive T cells decrease the odds of a mouse to belong to the good performance group. This population is also negatively associated with spatial memory function. In contrast, the proportion of CD8 positive T cells and also NK cells are positively associated with spatial memory. And no differences were found in other memory domains. And to explore if good and bad performers have different trajectories in the proportion of immune cells during aging, we performed again linear mixed models. But this time, knowing that males and females have such a distinct progression during aging as previously demonstrated, we decided to perform this analysis on males and females independently. And curiously, we observed that the few differences that we found were only present on males. In all the populations, females with good and bad performance present always similar progressions in the blood composition during aging. And regarding the males, I have here two examples. For instance, we observe that males with good performance in black present higher percentages of monocytes during aging, and they also present a decrease in the proportion of moderate decrease in the proportion of CD4 positive T cells during aging. And this association between CD4 positive T cells and cognitive function, even being moderate, is interesting because it reinforces what we already described and observed in healthy individuals. We reported that individuals with poor cognitive performance have higher effector memory CD4 positive T cells. In the last part of the work, sorry, it's not working, okay. In the last part of the work, we explored how plasma immune molecules correlate with age-related cognitive heterogeneity in healthy senior individuals. And even already described this association, most of the studies only report a small set of immune molecules, and a broader and comprehensive characterization was still missing. And to fill this gap, we used samples from an healthy aging cohort, same cohort that I previously mentioned in the introduction. And this cohort is composed by 190 participants, both men and women, with 51 to 87 years old. Those participants performed an extensive cognitive assessment. They performed several cognitive tasks that were carried out by trained psychologists. And those tasks were then grouped in different dimensions, in an executive function dimension and in a memory function dimension. And with the scores that those participants obtained on those dimensions, they were clustered and classified as good and poor cognitive performers. And this part of the work was done by other members of the team. And from those participants, we collected plasma, and using a multiplex bit-based assay technique, we measured 31 immune molecules. From those, only the immune mediators with more than 50% of the samples above the lower limit of quantification were considered for further analysis. And we started by comparing the concentrations of the immune mediators measured between good and poor cognitive performers. And we observed that the poor cognitive performers in red present higher concentrations of IL-1 beta, IL-8, IL-13, and TNF, and which indicates that the poor performers have a more inflamed phenotype. But with this kind of approach, we can't control for variables such as age and sex that we know that can impact this association. To control for that, we perform several regression analyses where we include on each model each immune molecule independently controlling for sex, age, education, mood, and also the intake of inflammatory drugs. And we use those models trying to explore which of the immune molecules are significantly explaining the variance of executive function and memory. In the case of the executive function, we found that IL-1 beta was the single immune molecule that significantly explained the variance of executive function. And it presented a negative association, meaning that higher concentrations of IL-1 beta are associated with worse executive function. Regarding the memory dimension, we observe that IL-1 beta, IL-1 receptor antagonist, IL-6, IL-13, IP-10, and TNF, all of them presented negative associations with memory function. We also explore which of the immune molecules significantly contribute to discriminate between good and poor cognitive performance. And for that, we use logistic regressions. And we observe that IL-1 beta, IL-6, IL-8, and IL-13, all of them significantly contribute to the discrimination between good and poor cognitive performance. And in this case, all of them increase the odds of an individual to belong to a poor cognitive performance group. And to sum up all the findings, in the first part of the work, we performed for the first time a longitudinal characterization of the proportion of the main blood immune cells. And with that, we observed that an overall increase in the percentages of the main innate immune cells and an overall decrease in the percentages of the adaptive immune cells. And we also reported an age-related sex dimorphism in the aging of the immune system in mice. By performing a multiparametric behavioral characterization in a large set of mice, dissecting different memory dimensions, we were able to characterize their heterogeneous cognitive performance and classify them as good and bad performers. We observed also that some immune cell populations at 18 months old are associated with their memory performance. Moreover, we also have shown a moderate association between the percentage of immune cells over time and cognitive performances in a phenotype that was only present on males. In the last part of the work, we have quantified 31 immune molecules in healthy senior individuals that present distant cognitive performance. And we have shown an association between peripheral immune molecules and cognitive heterogeneity in humans, not only confirming already well-described associations such as the IL-6, but also strengthen others like IL-1 beta, IL-8, IP-10, and TNF, and also revealing new players in such associations such as the IL-13. As future perspectives, and even not explored in this work, we think it's extremely important to extend this longitudinal characterization to cell function and cytokine production profile, because immune senescence is not only characterized by alterations in the proportion of cells during aging. It is also described to be associated with alterations in cell function, and it will help in wider our comprehension on the immune system aging. It is also important to measure serum inflammatory markers in mouse models and explore its association with cognitive heterogeneity. This part of the work is ongoing. And of course, it will be interesting to see if the profile in a mouse model, it is consistent or not with the results that we already obtained in healthy aging individuals. We think it's also interesting to explore and to correlate the blood immune molecules profile and with the inflammatory profile in several pain regions. And in humans, we intend to also to evaluate to what extent our findings are consistent in an independent cohort. And in fact, we are already establishing a new healthy aging cohort here at the ICBS. And last but not least, we think that it's really important in this type of study also to amplify the sample size and also to perform longitudinal cohorts that will allow us to contrast more robust and multivariate predictive models of cognitive aging that can be based on the characterization of the aging of the immune system. And I would like to acknowledge my supervisors, Margarida and Joanna, and all the colleagues involved in this work. Also the financial entities, and of course, not forgetting all the support during the last years from the neuroscience and the population health research domains. Thank you all.
Thanks, Claudia, for this interesting presentation. We have time now for questions. Any question? Claudia, I have a question regarding the task 3 and the definition of healthy aging cohort. What are the exclusion criteria? Because, for instance, joint pain or hypertension were an exclusion criteria. I'm imagining that, in fact, it is hard to find humans or men older of 75 years old without comorbidities and without any medication. So for me, as a doctor, it's interesting to understand what is health population at this year with this age.
Well, when we say healthy aging, we are also considering some age-related comorbidities that are not affecting their daily life. Of course, we are not including individuals with hypertension or diabetes. It's almost impossible to do a, let's say, pure healthy cohort. Otherwise, we will not have any individuals to follow. So there are several protocols, for instance, and criteria. At the beginning, they were really strict, but now the criteria are more abrangentes. Sorry, I don't remember. Okay. And we also include other comorbidities that are not affecting directly their life, their daily life. But in this case, we do not include, for instance, individuals with immune-related deficiencies, for instance, nor cognitive impairment. Let's say that these individuals, even though we discriminate them as good and poor, it's within the normal cognitive performance. It's just natural heterogeneity. Okay. And of course, we are not including other type of dimensions and not including depressed individuals. We control for mood, but even in mood, it's within normal ranges. So it's not, when we say healthy aging, of course, we are not being purist and not saying that don't have any kind of pathology, because it's almost impossible. Okay. But we control for main things that we know that can impact on such association between cognition and the immune system. Okay. That's why we also control for the intake of anti-inflammatory drugs. Even though it was a small proportion of the participants that were taking anti-inflammatory drugs, I think it's less than 15%. But even though we thought that it would be also important to control for that.
Okay, thank you. Ana Falcão.
Hi, Claudia. Thank you very much. I really enjoyed it. I was seeing your presentation and I was wondering, do you think this kind of phenotype of good and bad performance, at least the ones you had in mice, could it be reversible at some point? For instance, if you follow a cohort of mice and you divide them in good and bad performance, they don't have to be like super old, like the last time point. But do you think that if you put like the bad performance in like an enriched cage for months, they can eventually reach the level of the good performance? And of course, this would be great then to compare like the levels of cytokines and everything that you are analyzing before and after.
To do a protocol of enrichment, let's say cognitive enrichment, in at least in a set of the called bad performance, right?
Yeah, but not in a later time point, a little bit earlier. So just to see if they can, if it can be rescued, like with the environment enrichment or something.
with this work, the full characterization, as you mentioned, was only at the later time point, at 18 months old. But at the beginning of this study, we performed a pilot study in a small set of animals, and we characterized their performance at more or less at 11, 12 months. And at that time, we were also able to discriminate their cognitive performance. So maybe at that time, if we perform the cognitive characterization at 1 year of age, maybe we can then split the, let's say, bad performance and perform enrichment and see how it can affect, for instance, the immune system and their performance. But sometimes the problem is that by doing that approach, we'll have to perform 2 cognitive time points characterization. And sometimes it's difficult to repeat some cognitive tasks because they will not work. They learn and they will not perform when we repeat the task. Of course, we can think in an approach.
Even if it's 1 year later?
We can, sorry?
I mean, if you do it at 12 months, do you think like in 6 months after they can still remember the test?
Or remember, or even not only remembering, but act as a cognitive enrichment, but for all of them, for instance.
Okay, I see your point.
And then I can lose their variability later on. So I'm speculating.
Which could be good already. It could.
Yes, but with this approach, we'll not be able to see the variability that we want later on. Yeah, so it's interesting. We can think in using, for instance, a surrogate test, a single test that will be reflected in their different domains, for instance, and do a single, I don't know if it's possible or not, to help to segregate them, and then do a more comprehensive and full characterization later on. It's one possibility.
Yeah.
But
or
this
instead
kind
of
approach is difficult to do and to repeat tests. We need to be very careful when planning those type of experiments. But of course, enrichment, it's an interesting approach, and it's a different question, but of course an interesting approach to see if that will somehow modulate, because I think it's already described an association between enrichment and also the CD4 positive T cells and neurogenesis, for instance. I think it's already described. So maybe there is a good opportunity to follow on that approach.
So just one more very quick. I didn't see you mentioning in the end, but you are also planning to collect the CSF and do the same analysis as in the serum, or?
We have CSF from those animals. We will start over. We will start, but well, actually, we performed the serum characterization already in the mice, but I hadn't time to perform the fully statistical analysis. In the preliminary approach, I only observe a few differences, but I need to check. But we have CSF samples, but we didn't measure it yet, and we'll maybe, because the amount of CSF is too low, we will have to do pool of mice. So in that case, we need to think in advance which mice we are going to join, because they need to have a similar cognitive profile. So we have the samples to do that. We didn't perform that, so it's something that is interesting to see how the profile of the inflammatory profile in the periphery is related with the central nervous system in the CSF. That's the entire brain. So it's also an interesting question.
Or the meninges, right?
We have meninges frozen. We can extract RNA, for instance, and measure their inflammatory profile. And we have also the choroid plexus. So it's something that it's possible to do and to correlate, of course, with their cognitive performance.
Thank you.
Professor Gil Castro?
Okay, thank you. Cláudia, I have one or two questions. So you showed us a clear association with on good and bad performers with a certain cytokine profile, namely IL-1, IL-8, and so forth. So if you have these mice can you use these bad performers and modulate the cytokine profile, either neutralizing or stimulating or whatever, so that then you can try to convert them into a good performers? Or do you think that's not, because you're not killing the mice, right?
No.
So we are following the mice from, I don't know, day something to 1 year.
From 3 to 18 months old.
So if you have these mice, can't you try to manipulate this cytokine profile or other molecules to improve their, to check for their performance?
Yes, first, the immune-related profile that I described is in humans. And of course, and as you mentioned, we see an association between the profile between good and bad performers. Now we first need to confirm the findings in humans in mouse models in our cohort of mice, and see how similar it is or not from humans. If we find also in mice an association between, if we find in fact an association between the immune mediators and the immune mediators and their cognitive performance, we can think in advance to at least, for instance, thinking in the approach that Ana Falcão mentioned in trying to evaluate their cognitive performance and do environmental enrichment, but switching the environmental enrichment, for instance, at 11, 20 months old, and try to modulate their inflammatory profile and then see if they will improve their performance or not. So it's something that it would be interesting to see if it's possible to modulate or not. But thinking in the translational perspective, of course, we first need to think in approaches that can be translated later to humans. And sometimes blocking the full action of a cytokine in periphery, it needs to be done with careful, because it will impact also the immune response to pathogens and to other factors. So it's an interesting perspective to think in advance, to think in an approach where we can modulate the immune system, of course, to affect and to improve the cognitive performance of the mice that we know that will have a worse cognitive outcome later. So it's something interesting to think, of course.
Okay, so the other question, I don't know if I got it right, because I mean, it seems that I missed something here with mice and humans, but that's okay. So you showed us at the beginning that with age you have a decrease in percentage of in acquired cells, which is in line with the atrophy of thymus and bone marrow and so on. Do you have numbers for these cells?
If we try to assess the cell numbers, but we have a technical problem during the evaluation, and we had to switch the technique. At the beginning, we used one equipment, the Muse analyzer, and then we had to switch to the flow cytometer with beads to count the cells, and we had some variability between the approaches, so we don't trust on that counts over time. So we decided not to explore the cell counts. That's why I only showed a percentage of cells. So we try to evaluate cell counts, but due to technical issues, we were not able to do that and to
my question, can we associate this deficit in performance to the deficit of the immune system, or because you have the deficit in the immune system, it can somehow imbalance our relationship with, for instance, microbiome? And then what we see here is the result of this unbalanced equilibrium, and with consequence in performance. What I mean is that could this be a direct effect of the immune system or consequence of this imbalance? Yeah, what do you think?
with this type of approach, let me switch the slide. So with this type of approach, we cannot say which is the cause and the consequence, of course. But as you mentioned, we have the also you talk about the possible influence of microbiome on those associations, and
Well,
it's of course it's interesting. In this case, we don't know. It's interesting because a recent paper came out last week showing also the importance of immune surveillance in the near the meninges, in this case in the dural sinus that drain the blood from the brain, and they show that at those sites it was an important interaction between the immune system and the presentation of antigens from the brain. And we don't know if those cells will then go in circulation and of course have a different profile there, or and also if the profile in the periphery, for instance, that can be modulated by the microbiome, will affect their phenotype and will affect their function in the surveillance of the cognitive function. I think in this case we have a circle. I think it can occur both. I think it's the aging of the immune system that will alter their function. That can be modulated by several factors, diet, microbiome, and other factors, will affect capacity of cells to do the surveillance of the brain, and also the message that came from the brain and drained to those sites where the immune cells can communicate with the brain and to be presented to brain antigens will also modulate their phenotype. So I think both the periphery can modulate the capacity of cells to surveil the brain, and also the messages from the brain will also influence the phenotype of immune cells. And I think it's a two-player in this game. I don't think it's one exclusive.
Yeah, so one last question, if I can, Christina, can I?
Of course.
So, I don't know, for B cells, I don't know what you can do, but for instance, for T cells, you can do in an aged mice a transplant of a new thymus and have this side of the immune system to be recovered. So I don't know if something that you're thinking about or not.
Of course, that because one of the populations that always appear in human cohorts and also in mouse cohorts is CD4 positive T cells, and in this case we are showing alterations in terms of proportion of cells, not necessarily function. It's also interesting that we can stimulate those cells and see how their function are altered, but of course thinking in strategies to rejuvenate the immune system using, as you mentioned, in mice, using the thymic transplant can be a good approach, but in humans it will be difficult to translate the pure idea of a thymic transplant. But of course we can think in other strategies to do an artificial thymus and to able to rejuvenate the immune system. It's something to think in the future, of course.
Okay, I think we don't have more questions, so if you agree, we can finish now. Bye-bye to all and have a good week. Thanks, Cláudia.
Yeah, thank you. Thank you all for listening.